Hello, hello everybody. My name is Muhammad Farouk Sharifpour. People call me Farouk here, and I'm a veterinarian and a PhD in molecular parasitology. So the research that I'm going to discuss here for this evening is the first report of extracellular vesicle extracted from spirulina and its pro-inflammatory response, and hopefully potentially as a work as a vaccine adjuvant. So yeah, in here, this is a first report of such isolation and characterization of such EVs, and we were hoping to continue on this research for following years. Let us begin. Do you all hear me well? Just hear me okay, great. So what is spirulina? I don't know, many people nowadays are familiar with spirulina, and especially those who are keen in nutrition and athletes, because spirulina is considered to be a superfood, meaning that it has high contents of proteins, very have a lot of microelements and micronutrients like lipids, uh, uh, metals, essential metals, and antioxidants. So this spirulina is generally a cyanobacterium. It's generally a bacteria which is photosynthetic, and although the original size is a size of bacteria around 5 micron or 7 micron at some points, but they can form a back-to-back kind of formation, and they stick together and makes large filaments, and they have gliding motility inside water. This is my footage of spirulina. This is actually fast forwarded. I'm not sure if you can see it on Zoom. So now this is a fast forward motility. They kind of glide slowly inside water to sometimes reach for nutrients or different temperature. And this motility is derived by some specific proteins that will be important in this discussion in future slides. But yeah, they have very slight motility, and they kind of twiddle between each other to find better light source or temperature in water. So why it's called a superfood? Because it has a lot of good nutrients. So protein, it has more protein than anything that you can imagine. So we normally have this consider beef or chicken or soybean or egg as source of protein. Spirulina has way more protein than it has. That is why now they are seriously considering to use spirulina for space shuttles and space travels for astronauts, because aside from protein, which has it has other micronutrients that would help them to maintain their life in outer space. So with all this good stuff that spirulina has, it's called a functional food, meaning it has medicinal effects, or therapeutic effects, which we discuss now. But going a little bit back, so spirulina has been consumed by humans for a long time now. So over thousands of years history of spirulina consumption by humans in considering various sources that we found. But the nearest uh, thing that we found in Aztec people in today's Mexico, it's been said that in some books, a recorded history from 16th century, around 500 years ago, and it's been said in that history book that this substance, this spirulina, has been consumed by humans for a long time. and then in Central Africa for centuries as well. Like I said, it's a functional food, and there are a lot of studies these days that correlate the consumption of spirulina to various medicinal effect and therapeutic effect. The lot of protein discovered is going to work against hypertension, obesity, type 2 diabetes, and with lots of antioxidants is now proven that it works against cancer and various sort of colitis. So spirulina is farmed like other crops. It's either in small scale, you can have it in home in a fish tank with the right light source. You can grow it. It's something like that. And or in large-scale cultivation, you have now large spirulina farms which use natural or artificial light to grow them, or in like this photobioreactors in A, B, C, and in D, it's like open ponds. They grow spirulina. You might have seen actually there are lots of supermarkets, especially around Australia. They sell it as a powder form spirulina to add it to fruit salad or sprinkle it and various making smoothies out of it. So how about now we're going back to the EV world. So there is not much studies about cyanobacterial EVs. So we have lots of studies about eukaryotic originated EVs, even bacteria, but not cyanobacteria. There are probably almost 2 or 3 studies that's been done show studied extracellular vesicles from cyanobacteria, but none of them are from spirulina. Our paper is the first report of extracting EVs from spirulina culture. Cyanobacteria cell envelope is very similar to a gram-negative. It's actually if you stain it with gram stain, it will be gram-negative. However, with specific differences. So they have outer and inner membranes and a periplasm space, which resides those peptidoglycan macromolecules, and also surface layer proteins especially for motility. Also, like gram-negative bacteria, we have a lot of we have different forms of extracellular vesicle, which is to some point it's different than eukaryotic cells. So some of them we call it OIMV or outer inner membrane vesicles. OMV or outer membrane vesicle. This means that we have 2 layers of phospholipid membrane in gram-negative bacteria. So if the vesicle bud out from the outer membrane, they call it outer membrane vesicle. If the vesicle buds from both membranes, they call it outer inner membrane vesicle, which they have different mechanisms for doing. And there's a variety of shapes as well. They can be spherical, they can be oval shape, or aformal. With that in mind, we go straight to our research, that how we did extract spirulina and what adjustment did we use for doing that. And we introduced an optimized method. It's a combination of different old school isolation of EVs. And also we dig down to the molecular characterization of spirulina EVs and their cargo. And for the first time, we studied extracellular injection of this extracellular vesicle and exposing the mouse immune response, immune system to that spirulina EVs, and to study how it worked against, how it provoked immune system in mice. So spirulina culture. So spirulina in a lab environment is grown in something like this, a what we call it, a photobioreactor. I'm just going to change my laser point. Photobioreactor. So it maintains uh, uh, temperature, uh, agility, means it can shape or like orbital shape or make it, or using bubbles to agitate this spirulina culture inside it. And also the most important thing is lighting. So with that, for this research, we use cool white light or blue shifted light, 15 to 150 micro Einstein. And micro Einstein is a unit of light intensity. And we use air bubble and shaker, preferably shaker for that specific research, and a temperature between 20 or 30, mostly 26 degrees to be exact, but we used a different variety of sets. The mediums are consisting of different salts. We call it Zaruk medium or SOT, in which case we use Zaruk medium. It's probably the ingredient is a lot of probably 18 different salts and minerals plus nitrogen sources that we used to make this medium. It's a highly complex medium, but we had to make them in-house. So going forward, the photobioreactor is not fancy that we use. It's just what we use is something like this. I changed a bacterial photobioreactor into a homemade photobioreactor with an LED, adjustable LED light, which I could control it with my phone. And then the incubator has orbital agitation, so that's what we wanted. And then we use this fresh spirulina and grow this fresh spirulina until it goes to optical density of around 2.5, which is super high. It gets really green, blackish something. And then we process the sample, which I'm going to explain soon, and then fractionate the extracellular vesicles and characterize the size and concentration. Two methods, TRPS and a NTA, and also we characterize it with as a final characterization, we do cryo transmission electron microscopy. Yeah, a 2 liters of clone spirulina at OD 1. Sorry, the OD 680 1.3. It means that at a wavelength of 680 nanometer, we reached 1.3 of OD, which in that OD, spirulina gets so heavily cultured, and then supposedly the concentration of EVs gets very high. And then when for sample processing, in this sample processing, we took out the medium supernatant by filtering it through 0.45 filter systems, and then with low velocity centrifugation, several steps of low velocity centrifugation, 2,000, 4,000, and 10,000 Gs for 10 minutes. We kind of get rid of the debris or cell or larger particles that's in there, and or the remaining of the cells. And then we had several steps of filtrations using like Wattman filter, and then using 0.45 again for confirmation, and then we concentrated the supernatant because the volume is so high, it's about 2 liters. With the infrastructure and the equipment that we had, we couldn't do large volumes, so we concentrated with the cutoff proteins. And then we used a high velocity centrifugation or ultra centrifugation around 200,000 Gs. And before we saw we pelleted the extracellular vesicles, and they form like this brown pellet. Probably this pellet is from 500 mils of um, original medium supernatant. And why it's in a spirulina is green, but why the EVs are orange or brownish orange, that it boggled us for a while, but we realized that, you know, these EVs, it is spirulina on their surface, on their membrane surface, they have this beta carotene or beta carotene molecules that makes if they are extracted, makes them actually orangish brown. So then after crude extraction of that pellet, we went on to fractionation. So we did fractionation using size exclusion chromatography, and we found with this is ISO machine is AFC, automated fraction collector, and then we found and then we made 7 or 6 fractions out of it, at some points 9 even fractions, and we found that most of the EVs went into fraction 2, 3, and 4. This is because we speculated that this is because the majority of EVs are almost uniformal, and they have almost same size. I mean, the size distribution not that big, so it's actually small size distribution. That's why most of them went into earlier fractions. And then after that, we analyzed them with TRPS, which stands for tunable resistive pulse sensing. So it's a technology by ISON that using the ISON machine, the Exoid, which is really hard to work with, that measures the size and distribution of EVs by forcing them and passing them through a nanopore, a nano-sized pore. It's very clunky. You have to repeat it several times to get it right, but eventually it works well. So we found that the size distribution is almost, the mean diameter is around 100 to 130 nanometers, with a mode from 74 to 88 nanometer, and the concentration from for a concentrated extracellular vesicle suspension was around anything between 5 to the 11, sorry, 1 to the 10 to the power of 11, and to power of 12. That's very huge numbers of extracellular vesicles. But we did a confirmation with NTA, good old NTA. NTA is very easy to use. In 10 minutes, you can read everything. However, it might not be that accurate, especially in the size distribution department. So for the concentration, we found the similarities between the two devices. The concentration is to the point of to the power of 11 here as well. However, for the size distribution, it's completely off. I mean, it shows it way more than it should be, and it shows around 300 to 2 to 300 in diameter. How do we know that this one is not right? Because for the final assessment, we did cryo TEM. Sorry if it's TEM here, it's actually we use cryo TEM. And yes, there we go. We have a lot of EVs. And then if we go to a higher magnification, you can exactly see the boundaries of EVs, the two layers of uh, 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 and the two layers of phospholipid membrane, and also uh, the surface molecules or the crown. So, and with this, we compared the sizes with the two machines. We realized that the sizes are much closer to the result of TRPS. So, and then we found different shapes of and different forms of EVs in there. So we found these outer membrane vesicles, small and large, and this inner outer membrane vesicle or outer inner membrane vesicles in that. And we have also different variety of exotic shapes of extracellular vesicles. My computer seems to be frozen. Apologies for that. Yeah, getting better now. So I'm just gonna summarize it here and just go into the next step, which is immune response to spirulina-AVs out of sheer curiosity. And we had the facilities to do the animal experiment. We exposed mice to injected spirulina-AVs. And for the beginners, we did a few pilot trials and with few mice, and we saw that actually it increases the uh, almost every time, and we repeated that several times with different batches of spirulina. It always increased the percentage of neutrophils to the site of injection a lot, while keeping other and also M1 macrophages. So two cells are dragged into the site of injection. One is M1 macrophages and then neutrophil. And in peritoneal exudate, we did inject an IP. In peritoneal exudate, we found these two cells are white. But then we didn't we were not convinced. We continued on. We used a long-term exposure, long-term administration of spirulina-AVs to mice to see what happens. And then we did use several kind of several injections, 4 injections before measuring before measuring immune response to see if still works in long-term exposure. And there we go. We still had it, neutrophils, and neutrophils are so high, and also this time interferon gamma Th1 cells also are up so much. However, being said, we did not see any adverse effect in mice. For example, if it was something really pro-inflammatory, it should have some adverse effect in the site of injection or in overall health of the mice. We did not see anything that comes. So it got us in thinking that maybe this has a safe pro-inflammatory response that we can see in mice. And we actually measured a lot of stuff, but this is the highlighted results that I'm showing you here. And it got us thinking, maybe this is something that we could use as an adjuvant. Maybe this is because not every pro-inflammatory response is safe, but this is a cell-specific pro-inflammatory response. It doesn't actually have any toxicity. It doesn't have any other adverse effect of pro-inflammatory response. So we thought that maybe it could use as an adjuvant. And centrally, we did that experiment. We had this experimental vaccine antigen in our lab against a flu called schistosoma hematobium. And we had spirulina-AVs. So as a result, we combined them together and injected them at the same time to mice, subcutaneously this time. So the site of injection changed as well. And surely we found that that specific IgG antigens against schistosoma hematobium increased by 10,000-fold, by 1,000-fold compared to the antigen itself. And it's much more comparable to a commercial alum CpG adjuvant. So it means that we found a biologically active adjuvant, which is non-synthetic, actually coming from nature, that can induce, they can boost the immune response against a specific antigen while we combine them together. So in this experiment, we also added a non-spirulina extracellular vesicles as a control. Well, I did a lot of research on orange extracellular vesicles, so I extracted so many times as a control. Now we have it's orange extracellular vesicles here as a control. You see that there is no difference. So it means that no difference between here and the one that we didn't use any EVs and PBS for that matter. We see that this is specifically can be used as an adjuvant. Then again, I'm just leaving summarized here. I don't want to go into details. However, why this would happen? One of my colleagues asked me that why do you think an extracellular vesicle from a species called spirulina should have that effect? And my response was, why not? I mean, we have so many strange things in science. Why not this one? And with that in mind, we wanted to know how actually it happened. Why this spirulina should have such effect? And bearing in mind that spirulina itself, I mean, intracellular capacity space of spirulina has anti-inflammatory effect in many diseases because it has antioxidants and so many other proteins inside spirulina cell. But extracellular vesicles of spirulina has pro-inflammatory effect. That was really interesting for us. And we went down to proteomics. So that's the thing that we had capacity in our lab, so to do proteomics. And after proteomics, we found 45 unique proteins in the spirulina EVs, which means that it not specifically means that these are cargo, but actually cargo and surface proteins. So we found 54 unique proteins with different biological kind of, we predicted those biological function. So if you said most of them are related to photosynthetic and electron transport and some other organonitrogen compound and carbon fixation. Also, we found some structural proteins or cellular components like outer membrane. Some of them were referring to photo against extracellular region transporter proteins or photosystem again. And some have molecular function and enzymatic function like transferase and anion binding enzymes and so on. And we do a protein family, and we found those 54 proteins that belong to 7 major superfamilies of proteins. And interestingly, in the literature, we found that 3 of these superfamilies can have pro-inflammatory response upon injection in mice. And again, S-layer protein, which is will be important in my future slides, again, is one of them. So in summary, up until now, we what we discovered is that is first a new way, a combination of filtration and ultracentrifugation, and size exclusion chromatography to extract spirulina EVs. And we characterized it with TRPS, cryo-TEM, and NTA as well, which is not shown here. And we also treated mice with spirulina EVs that showed very sharp pro-inflammatory response by increasing neutrophils, M1 macrophages, and interferon gamma Th1 cells. And we found that it can have adjuvanticity properties by increasing the IgG level in response to the antigen that we injected with spirulina EVs. It increases the IgG response by 1,000-fold. And we found superfamilies using proteomics, and we found 54 proteins in 7 superfamilies, that 3 of them can actually have pro-inflammatory effects upon injection. What now? So why is it important? So it is important because nature every time, all the time, give us something to work with. A new way of production of adjuvants is coming, a natural way. We need to still do some further safety studies if we don't have active grant at the moment, but future studies, if we get some grants, we'll be able to do some safety studies with the infrastructure that we have and the equipment that we have in the lab. But for vaccine application, we have some ideas which we put forward a grant. It was not successful, but we hope that we can reapply at some point to get some to get some great ideas. And this idea was production of vaccine and antigen inside spirulina as an integrated way to be a more cost-effective and financially efficient way of making vaccines. How does it work? So it goes back to one more thing that we did in the lab, which is successful gene editing tool in spirulina. It has been done recently in the United States, especially a company that they did find a very nice way and easy way to do gene editing inside spirulina. However, we established in-house by swapping native genes, native but not lethal genes with green fluorescent genes, just as a marker to see if we were successful. And surely we did establish a successful transformation in spirulina, which we express transgene EYFP, or enhanced yellow fluorescent protein. As you can see here, we established it by swapping EYFP with a native gene, which was not, removing it was not lethal. So our future direction would be actually anchoring a specific antigen. I don't want to name it because it's a kind of confidential, a specific antigen of a disease to surface proteins of spirulina, which ends up in the extracellular vesicle. In that way, one spirulina can be a mini factory that you can expand, culture, and mix it tons of these EVs to be used as vaccine and antigen. So some people call it self-adjuvanting vaccines, but actually it's not the right term. I would say it integrated vaccine antigen production rather. So why we use spirulina as my last slide? I hope that I didn't talk too much, but as why we use spirulina for such idea. The first reason is it's photosynthetic. It doesn't require that much energy. It doesn't require that much infrastructure to give us energy, and it's also readily available. It's non-toxic. It's edible. And it's very low cost and hyper scalable culture. So you imagine that you can have one spirulina that is transformed and genetically engineered to make that EVs that's also engineered, and then you can culture it all the way you want with lots with it's photosynthetic. It doesn't require that fancy medium, which is very expensive, and you can grow it in seawater. And it's also spirulina is proven to be very efficient in expressing transgenes if it's established. So, and with that in mind, spirulina can be what I call it an efficient, hyperscalable microfactory fueled by sunlight, meaning that you transform one spirulina, genetically transform one spirulina to make it your product, and then to somehow anchor it with your extracellular vesicles, and you will have a factory of making vaccines with a cheaper cost, way cheaper cost, especially for developing countries. Well, I hope you enjoyed my research. So I would like to thank my very best colleagues in here, Dr. Suchandran Sikter from James University, Yeda Wang, Professor Alex Lucas, my supervisor, Professor Jamie Seymour, Dr. Nana Quickman from UQ, University of Queensland, Dr. Robert Courtney and Tamara Thomas, and also many other people that helped me achieving the results, and also the National Health and Medical Research Council of Australian government to provide us with the funding, AITHM and James Cook University, and many others that we couldn't really mention them here. Well, thank you so much. If you have any questions,
Thank you, Farouk, for your presentation. It was very informative, and thank you for the background about spirulina. This is definitely a very sustainable source of extracellular vesicle. Yeah, we ran a little bit long, but I have time, 5 minutes for questions from the audience. I also have a question myself. So first from the audience, if there is any question, this is your time. Okay, I think I will go ahead with my question. So first of all, is especially talking about the characterization, the proteomic characterization. You talk about 54 unique protein. This means 54 protein, they are only in the EV and they are not present in the source, in the spirulina itself.
No, the term unique here actually it's a proteomics kind of terminology. It means that these protein are you have kind of what do you call it? You have 54 unique hits in when it comes to the mass spec with proteins. 54 unique hits and sequences that comes out of this. It means that there is at least 54 different proteins in that regard. In 54 different proteins can be extracted from the EVs. That's what it means.
So that's
and those EVs, those proteins, most of them have orthologs or analogs in other cyanobacteria or other gram-negative bacteria as well, but the sequences is slightly different.
But this leads to the second question I have. Actually, why use the EV instead of using the spirulina itself? Have you tried to compare the adjuvant effect between the two?
That's a great thing to ask. There are several things to be discussed here. One is spirulina is a big cell, big long filaments, right? And injecting spirulina itself might not be
well.
very
I mean, even from the tissue, you can simply process the tissue and make like a cocktail of all the components of the tissue.
Yeah, homogenize. But remember,
homogenization of the tissue.
Yeah, remember I discussed in one of the slides that spirulina itself is anti-inflammatory, has anti-inflammatory responses. the spirulina cells, and it's been used in colitis model in mice that it actually reduce inflammation. And it's been done before. However, the extracellular vesicles represents not the whole cell, but actually some parts of cell. It's just the outer membrane of the cell. That's what we're interested in. That's what we use the extracellular vesicle. We didn't actually choose the direction that we took. We just wanted to report extracellular vesicle from spirulina and do a proteomics to send a cargo. But by a happy accident, we used it in mice and we saw the results. That's why we took that direction to adjuvanticity. And it was actually makes it more important than it was before. And you know, as a technical kind of point of view, in the near future, the EVs will be licensed to be injected or to be consumed as general therapy or parenteral injection. It's not approved yet, but it will be. There's a good chance. So that's why we thought of this injectable material using EVs, these nanoparticles that you can deliver something into the body, or you can have some therapeutic effect.
There is a question from the audience. I'm going to read it. Do you add antibiotic in your culture of spirulina?
Well, that's a great idea. Yes, we did. So the spirulina culture, it's normally when you receive it, I imported it from Japan. This specific batch, actually we acquired it from inside Australia. So I cloned it first. So it's not axenic. It's not pure. They call it xenic culture. It means it has other bacteria in it. So for that, I had to clone it, meaning that I had to purify the spirulina culture so it's be uniform, one cell, one type of cell, and get out the others. One of the stages, I had several stages. One of the stages using an antibiotic that spirulina is resistant to naturally, which is kanamycin. Spirulina naturally resistant to kanamycin. So we added kanamycin as one of the steps of purification of the spirulina. Other than that, it was specifically in sterile condition, the culture, and also spirulina itself, when it reaches a certain OD, it kills all the other organism, and the reason being it makes the medium so high pH that nothing else can grow in.
That's very interesting.
I hope that answers the question.
Yeah, I think you did. Yeah, you did. I have another question. Actually, I was surprised about the color of the EV, which are kind of orange compared to the algae, which is green. You explained because they are rich in carotenoids, right?
Yeah, exactly. So we were surprised too, and then I dug literature, and then apparently that's it. I mean, if you look at the surface of this specific spirulina or other cyanobacteria as well, it's full of carotenoids, and then it makes it orange, and sometimes it's brownish orange. And there are other, just one more study that I can think of from memory, I'm saying, that actually extracted EVs from another cyanobacteria. It was also orange. So it has actually confirmed that, yeah, this is EV from real spirulina.
Well, probably that's the main component for the photosynthesis and photoprotection of the algae, I guess, since they are rich in carotenoid. I have one last. Oh, probably there is some question from the. Yeah, I have a question from the chat. So from Twang, which specific protein among the 54 identified in spirulina EV are responsible for activating immunoresponse, and through what cellular receptor and signaling pathway do they function to produce the observed pro-inflammatory and adjuvant effects?
Good question. I'm not sure if I'll be able to answer that question precisely, but I remember that we have several hits of protein, 54 unique proteins, but those 54 proteins are classified or categorized into 7 superfamilies. So seven superfamilies, or they call it PFAM, protein family. Not superfamilies, families. They're different in terminology. They call it PFAM or protein family chart. In the PFAM chart, there are 3 different proteins. If I'm still sharing, I'm gonna go back to the specific slide to explain. Yeah. So in here, you can see RTX calcium binding nanopeptide repeat. Okay. This is one of them. And then S-layer homology domain. These S-layer proteins are a lot of various proteins that help the spirulina to make them move. And carbohydrate selective porin, or OPRB family, is the other one that we found at least these 3 proteins, all of them. We found evidence in the literature. Although in other bacteria, not in spirulina, they can have this pro-inflammatory response. But through what pathway, I'm afraid I don't remember that. So I have to go back and look it up.
Okay. I have one last question because, this is kind of a hot topic, the EV corona, So I believe the corona may affect also your study. And what do you think about how the corona can actually affect the adjuvant effect of the spirulina EV?
Sorry, can you please repeat it?
Yeah.
It broke up for a minute.
So as you know, the corona is now a hot topic in the EV field. I believe more or less you thought already about how the corona can affect the adjuvant effect of the EV. It's really depend on the source, how isolate your EV. It's kind of a biofluid, may really affect the EV biology itself.
Yeah, it does. And definitely, not I'm sorry, I'm going to answer your question, but I'm going to give a small background. So not all isolation methods can rescue the corona, right? Some isolation methods can actually destroy the corona.
Yeah, that's what I mean.
Depends on this format. Yeah. And we were fortunate to do cryo TEM, we see that yes, we still have intact corona around our EVs. And I reviewed probably more than 100 cryo images, and I noticed that not all of the EVs have corona. So it means that in the process, some of them, they lost their corona, right? And yeah, and we found that actually all of these 3 proteins that you see here are corona proteins, are surface proteins. They're not cargo. So that's the good thing that we found. The S-layer protein definitely a surface protein. We know that because it's a motility protein. Spirulina can do its gliding motility through these S-layer proteins. Porin as well, carbohydrate selective porin as well. This is as well as a channel kind of protein. Is just a channel. So these are all surface proteins inside spirulina, on the spirulina EV, and it kind of gave us the reassurance that we did do a good way of isolating the EVs because we still have intact corona in them. But yeah, I'm sure we still don't know many things about the corona. First, are these EVs are uptaken by those cells by I'm sure neutrophils or and macrophages are, phagocytosic cells, right? They do phagocytosis. But we don't know if just contacting and then touching and exposing to EVs can cause them to react via the surface proteins, or they has to be ingested. It has to be phagocytosed and then make their effect. We don't know that because we didn't actually get in time and we didn't have enough resources to continue on the research. But for future studies, we need to consider doing those stuff. So we have some enzymes that, enzymes specifically in a prokaryotic cells. We have all enzymes, DNA, everything inside the cytosol of bacteria that they may end up inside the EVs, right? We haven't really went down to which protein exactly are doing. We need to purify each protein to see which one actually caused this thing to do the mechanism of action. Sorry, I went overboard a little, yeah, but I hope that you get the overall message.
Yeah, it's okay. So I think thank you very much for your presentation. It was very, very informative, and we look forward to see the next chapter about spirulina.
My pleasure.
There is still a
Thanks
lot
for having me.
Thank you. And see you in 2 weeks with the next EV Journal Club. Thank you again, Farouk. Keep in touch.
Thanks for
Bye-bye. Thank you, everybody.
Thanks.